Hey everyone, this is Mrs. Mallard, and this lesson is on multiple alleles, specifically the example that we're going to be going over in this lesson is on blood types. So today, after this lesson, you should be able to identify the 4 different blood types. You should also understand which blood types are called our universal donor and which blood type is called our universal recipient. And the other thing that we're going to get additional practice with is doing Punnett squares with blood types. So first of all, before we actually get into the blood types, I wanted to just review the word allele. So allele is just a different version of a gene. So here, whatever this particular trait is, maybe it's flower color, for example. We have one allele represented by capital A. Maybe capital A is for white flower color. And then the lowercase a over here is a different version of flower color, and maybe lowercase a represents purple flowers. So 2 alleles, a lot of the Punnett squares that we have been working with, we have been working with 2 different alleles, capital A, lowercase a. Today, we're going to be looking at blood types, which is an example of multiple alleles. So what this means is that instead of having 2 different versions or 2 different alleles like capital A and lowercase a, in this example, we actually have 3 different alleles. So notice that we've got 3 different alleles. One allele is going to be represented with a lowercase I. And then we have 2 other alleles. Notice that these both have a capital I, but we have to distinguish these 2 by putting a capital A like a superscript. And in this one, we have to put a superscript, but we're using a capital B. So in this instance, I have 3 different alleles. Of course, my lowercase I is my recessive. And then I've got I, capital A, I, capital B to represent these 2 other alleles. So we have multiple alleles represented here on the board. Now, what I'm going to do is I'm actually going to come out of this because we're going to talk about the 4 different blood types. So there are 4 different blood types. I don't know if you all know what the 4 different blood types are, but we are going to I'm going to go ahead and pick red here. Okay, there's 4 different blood types. We have folks who are type A. We have folks that have type B blood. We have folks that have what's called type AB, and we have some people who have type O blood. So those are the 4 different blood types. So if you don't know what your blood type is, you are one of those 4. And so these are the 4 different blood types. On the other side, what I'm going to write are the alleles or the genotypes for each particular blood type. So for type O, a person who has type O is going to have 2 lowercase i's. So you can kind of see type O is kind of serves as our recessive. If someone is type AB, they are going to have 1 allele for type A blood and 1 allele for type B blood. And notice that because they have 1 of each, we say that they have type AB blood. All right, now with type B blood, we can have people who are homozygous, which means that they have capital IB, or we have folks who can be heterozygous, which means they have I, capital B, lowercase I, like that. All right. For type A blood, we have the same situation. Someone can be homozygous, which means they would be IA, or they could be IA, little I, kind of like with type B there. So these are different phenotypes. So people can either be type A, type B, type A, B, or O. These over here are the genotypes. So these over here, this is what we're going to be using when we work out Punnett squares for different individuals. All right, this we would say is homozygous type A blood. This is heterozygous type A. This is homozygous type B. Or excuse me, this is heterozygous type B. This is homozygous type B. AB, it's just this. If someone is type AB blood, this is their genotype. If somebody is O, they're going to be two lowercase i's. Okay, so let's go ahead and let's go back. Let's go back to our PowerPoint here. All right, so this is what I just went over here, the different genotypes and then the different phenotypes that a person would have. All right, so let's take a look at what does all this mean? The type A, type B, type what exactly does that mean? So on your red blood cells, there are carbohydrates that stick off that are called antigens. They are found on the surface of the red blood cell. So your blood type is determined by what antigens you have sticking off your red blood cells. So here we're using kind of these green spikes to represent A antigen. So if you have type A blood, you have A antigens that actually stick off of your red blood cells. If you have type B blood, we're using the little circles to represent B antigens. If you have type B blood, you have B antigens sticking off your red blood cell. Those of you who are type AB, you have a combo. You have a combination. You have A antigens and you also have B antigens sticking off your red blood cells. And then if you have blood type O, think of O as in no or O as in zero, meaning that you don't have A or B antigens. You don't have either of those. So you have no antigens sticking off your blood type. So think of it as no or like zero antigens. All right, so that's what determines your blood type are these antigens that are sticking off of your red blood cells. Now, the other thing that is that we need to talk about are antibodies. So antibodies, these are proteins that circulate in your, that are part of the immune system. And what they do is they will react when they come in contact with an antigen that they do not recognize. So obviously you are born with your red blood cells have a specific antigen on them, but you also have antibodies as part of your immune system that will react with antigens on red blood cells that your body is not familiar with or does not recognize. All right, so we're going to we'll kind of talk about how these two play together here in a moment. So I have this chart, and this chart really kind of summarizes everything, everything with blood types. So remember, if you have type A blood, you have these A antigens that stick off of your red blood cells. If you have type B blood, you have B antigens that stick off your red blood cells. If you have type AB, you have a combination. You have some A antigens and you have some B antigens. Remember, if you have type O blood, you do not have A or B antigens. So you have no or zero antigens sticking off your red blood cell. Now let's talk a little bit about which blood types can donate and receive from other blood types. So let's take a moment there. Notice folks who have type A blood, they have antibodies against B. All right, so here it says antibodies present. So what this means is if I were to take this red blood cell and if I were to put it in someone who has type A blood, these antibodies are going to actually attach to the antigens that stick off of the blood cell here. What that is going to cause is it's going to cause clumping in the blood. We do not want that. We do not want clumping in our blood. So that is why we cannot give someone who has type A blood, we cannot give them type B blood. Now let's do the reverse. if we have someone who has type B blood, if I try to put A blood into that person, notice they have antibodies against A. So what this means is if I put this red blood cell in this person, the antibodies are going to attach to the A antigens, and that's going to cause our blood to clump. All right, clumping, we don't want that to happen, so we cannot give a person who has type B A blood. All right, now let's look at someone who has type AB blood. Notice that there are no antibodies against the A or the B antigen. All right, they do not have antibodies against the A or the B antigen. Some students say, wait a second, they don't have antibodies. Do these people get sick? No, 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 no. We are just specifically talking about antibodies that would attach to the A or the B antigen on a red blood cell. All right, so people who have type AB blood, they do not have antibodies against the A or B antigen. And it makes sense because look, they have A antigens and they have B antigens on their red blood cells. So you don't want any antibodies that would attach to these antigens because that means that their blood would clump. So because of this, because people who have type AB blood do not have these antibodies, they are able to receive from any blood type. So if I put type A into this person, there are no antibodies against A. If I put type B into this person, there are no antibodies against B. Of course, type O, they don't have any antigens anyways. So if I were to put this blood cell into this person, it would work. So type AB individuals are called the universal recipient. They can receive blood from anyone. And the reason why they're able to do that is they do not have antibodies. All right, let's take a look at group O. We know that group O does not have any antigens on the red blood cells. So if I were to put O into someone who has type A, I don't have any antigens on my red blood cell for the antibodies to connect to. Same thing if I put type O into someone who has type B. There are no antigens for these antibodies to connect to. So I can put O into group B. I can also put this red blood cell group O into someone who has type AB, and that will work just fine because remember, type AB is our universal recipient. They don't even have any antibodies. And the other thing is that type O doesn't even have any antigens. So type O is what's called our universal donor. Type O can give to any blood type. Now, let's take a look and let's see who O can receive from. So notice type O has antibodies against A. So if I were to put a red blood cell that was type A into group O, these antibodies are going to attach to these antigens. We're going to get clumping. If I were to put type B blood into someone with type O, I have antigens against B. So these antibodies are going to attach to these antigens. We're going to get clumping. The same thing is going to happen if I put type AB blood because the antibodies against A are going to attach to the A antigens and the antibodies against B are going to attach to the B antigens. So we're going to get clumping there. So the only blood that someone who has type O can receive from is another person who has type O. All right, so just to kind of summarize this, type O is our universal donor and type AB blood is our universal recipient. So we keep talking about what happens if you give someone the wrong blood type, and what happens is the antibodies connect to the antigens and we get what's called agglutination, which is just another term for saying that the blood will clump. So if you notice, I have a sample here, and if you notice, the blood looks kind of grainy looking. That tells us that we are getting clumping. So this is nice and smooth. This is no clumping, and then the graininess here indicates that we are getting clumping. And we don't want the blood to clump because that could possibly be fatal. Now, the other question that students always ask is, well, what's the deal with the positive and negative? I'm O positive, or I'm O negative, or I'm B positive, or I'm B negative. So what's the deal with the positive and the negative? So the positive and the negative, there is an extra antigen. It's called the Rh antigen. So we've already talked about if you have A antigens, you have type A blood. If you have B antigens, you have type B blood. Well, if you are positive, you have an extra antigen called Rh, the Rh antigen. If you are negative, that means your red blood cells don't have this. Okay, so this is just an extra antigen. Positive if you have it, negative if you don't. All right, so this is our last slide. So another question that students always have as well is my blood type rare? Is it common? And so this is just kind of a distribution of how many, kind of looking at the population. Notice that we have a lot of individuals who are O positive. All right, so a lot of folks who are O positive. Also have a lot of folks who are A positive. And it looks like if you are AB negative, that's the smallest percentage of the population. But you also have to remember that if you have type AB blood, remember that you are a universal recipient. So you can get blood from type AB, and O individuals. All right, just the last kind of thing to wrap up. So knowing your blood type, the main thing that it's used for is blood transfusions. You want to make sure that if someone is getting a transfusion, whether it's they've been in surgery and they've lost a lot of blood, or maybe through childbirth, they've lost a lot of blood, or maybe they were in a car accident and they lost a lot of blood and they need to replace that blood so their body doesn't go into shock. You want to make sure that you're putting the right blood into that individual because you don't want that clumping to occur. This is a little kind of less common now, is you can use blood tests for paternity testing, but we already talked about in the biotechnology unit that DNA is often more used for paternity tests now, versus blood types. But these would be two reasons why you would want to know your blood type. So our next thing that we are going to do, now that we've learned about blood types, is we are going to get some practice using blood types in Punnett squares.